Hey everyone, Professor Davis here, and we're going to dive into the second part of chemistry for biology, where we're going to be looking more at the organic chemistry side and really focus in on those biomolecules that are going to be important. So when we look at this idea of the fundamentals of organic chemistry, we need to remember that all living things do contain 4 types of organic molecules. These organic molecules are also known as biomolecules, bio meaning life. So these are molecules that are really important for life. And these include things like carbohydrates, lipids, proteins, and nucleic acids. Now we are going to talk specifically about each of these in this particular video. So with organic molecules, we do see that they are associated with living matter. Now, this does not mean that inorganic molecules are not important to living things and living cells, but we talked more about the inorganic parts that are important for living cells last video. Now, organic molecules do contain 2 things, and it's important to note. In order for a molecule to be considered organic, they have to contain both carbon and hydrogen. Okay, not just one, they have to have both. Some examples here are hydrocarbons. This is an octane that's found in gasoline, and you will see that it has what we call a carbon backbone. So it goes all the way across with carbon, and then it's covered with hydrogens. Okay, and so carbon is an essential ingredient in all of the biomolecules we're going to discuss. So because carbon needs 4 electrons to complete its outer shell, because remember we talked about bonding last video, it will share with many different things. It could share up to 4 other elements like we saw with hydrogen. This allows for a lot of variety when we're talking about a carbon backbone. Carbon can bind with itself as well as things like nitrogen, oxygen, phosphorus, and sulfur. And remember, these are the elements that are going to be really important to living things. And if you can remember the acronym CHNOPS, okay, carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur, that's going to be helpful. Now, remembering our rule that we just talked about, where organic molecules have to have both carbon and hydrogen, let's see if something is inorganic or organic based on its chemical makeup. So our first one here is H2O. This is water. It does have hydrogen, but it lacks carbon. Therefore, it is inorganic. We also see the next one is C6H12O6. In this particular case, we're looking at sugar or glucose. This one has carbon and hydrogen, so it is an organic molecule. The next one is CO2. This is carbon dioxide. It does have carbon, but it lacks hydrogen. Therefore, it is inorganic. Table salt is next, NaCl. This one doesn't have carbon or hydrogen, so it is also inorganic. And last but not least, we have our methane. This is CH4. It does have carbon and hydrogen, so it is organic. So make sure that you can tell whether a molecule is organic or inorganic based on its chemical composition. Now, organic molecules typically are a lot larger molecules. We call them macromolecules because they're very big. And they use mostly covalent bonding. All right, so we're going to see covalent bonds present, and they are going to make very large molecules that we call macromolecules. Now, functional groups are a big part of this, guys. Now, functional groups are atoms with specific chemical properties, and they have a very consistent behavior. So regardless of what they're attached to, like which carbon skeleton they're attached to, they're going to act very similar, no matter if it's part of a carbohydrate, a protein, a nucleic acid. They're going to have some similar properties. Now, these carbon skeletons are going to be that carbon backbone we talked about, and some functional groups are going to have certain characteristics like being polar, where they may have more of like a positive charge or maybe a negative charge. Sometimes they're acidic, sometimes they're hydrophobic. It just depends. They're going to have some of these different types of characteristics. So a single macromolecule may contain a lot of different functional groups, and this will make certain parts of the macromolecule have certain properties. So the R stands for the rest of the molecule or the carbon chain a lot of times when we look at this in a kind of picture sense, and only the functional group is involved in the reactions. Okay, and so this is going to be really important because those functional groups are going to determine how that macromolecule may act. So if we look here, you can see that you can have a long carbon backbone. That's the R part. It could be super long or short, doesn't matter. And then a functional group is attached. That functional group is going to give us different types of characteristics. Now with these functional groups, these are just some of the more common functional groups that are out there. I don't expect you to like memorize these, but if you see them, there are going to be certain characteristics present there. So hydroxyl is an OH. This is an alcohol and it's present in some sugars and some amino acids. We also have a carbonyl group. This is going to be where you have a double bonded oxygen and a hydrogen in some cases, and that's an aldehyde, and these are present in sugars. If it's just a double bonded oxygen, we call that a ketone. These are also present in sugars. You probably have heard of ketones, especially if you've heard about the keto diet, all right? But these are associated with those carbohydrates. Carboxyl groups have an acidic type of characteristic, and they're a double bonded oxygen with an OH. These are present in fatty acids and amino acids. Amino groups are going to be a nitrogen with two hydrogens. These are also known as an amine, and they are present in amino acids. And amino acids, we'll learn in a little bit, are the building blocks of proteins. We also have sulfhydryl group. The sulfhydryl group is going to be where they form a lot of disulfide bonds, and we do see these present more in amino acid structures for proteins. Phosphate's the last group, and when we look here, these are going to be present in nucleotides and phospholipids. And so these are going to be really important for like DNA and RNA, as well as the components that make up your plasma membrane of your cells. So some functional groups allow molecules to react with water, and sometimes they like water and sometimes they don't. So why is it important that some biomolecules are able to react with water? Well, the big thing is if they like water and they are hydrophilic, this is important because we are made up of 70 to 90% water. Our cells are made up of 70 to 90% water, which means these molecules will need to be able to react with it. So the ability to interact with or be soluble in water, profoundly affects the function of an organic molecule. Remember, there's two terms that we've discussed already. Hydrophilic means they're soluble in water, means they like water. On the other hand, if they're hydrophobic, they're not soluble in water, and this is why they call them water-fearing. They do not like water. Something we need to look at when we're looking at a lot of these different large molecules are what we call isomers. Isomers are molecules with the same molecular formula, but are structurally different. So this would be like if I was to give you a bag of Legos and you had the same colors and the same exact type shapes of pieces as the person next to you. So every bag of Legos was the exact same. However, I didn't give you instructions on what to build. I just said build something and use every block. What you build is going to look totally different than the person sitting next to you, because even though you had the same parts, you had different ideas of what you wanted to build with it. We see the same thing here with isomers. If you look here, iso means same and mer means parts. So they have all the same parts, but they may look very different. This leads to variations like structural differences, which then ultimately lead to functional differences, and it makes diversity possible. So here are some examples here. In this particular case, both of these molecules have 4 carbons. They also have 10 hydrogens. So their formula is C4H10. But if you look, the way that they put them together and they link together are very different. So you have butane, which is all very structural in one row. And then you have isobutane that actually kind of comes up and then across. Okay, and so the structure is going to be put together differently, even though the parts were the same. Another really good example of this is with our basic sugars, C6H12O6. So here they each have 6 carbon, 12 hydrogens, and 6 oxygens, and they could be, based on how those parts are put together, either glucose, fructose, or galactose. Some of the differences can be very subtle, like with glucose and galactose, or they can be very different when you look at fructose. All right, and so this again is an isomer. Now, when we're building these very large organic molecules, we're going to see that it's going to take a lot of small parts put together to make them. So the large organic molecules a lot of times are going to be abbreviated as LOMs, and the small building blocks that make up those molecules are going to be the small organic molecules, or SOMs. Okay, and so when we look at this, the monomer is the small side. Mono means one and mer means part. And so this is going to be the building block. This is going to be what we want to put together in order to make the large molecule that we call a polymer. Poly means many, mer means part. And so these are the large molecules that we also call macromolecules. All right, so I have a little chart here to look at those biomolecules, and we want to look at what they are when they are the polymer side, as well as the building block, which is the monomer. So our first biomolecule here is going to be carbohydrates. Carbohydrates can be complex carbohydrates, which we call polysaccharides. Again, poly meaning many, saccharides means sugar. On the other hand, the monomer, okay, the building block of this is going to be the simple sugar, or what we call a monosaccharide, one sugar. Fats are going to have this ability to be called lipids. These lipids generally mean fats, oils, or waxes, so there's different types. The building blocks, though, for these, the monomers, are going to be fatty acids and glycerol. The way those are put together determines if we're looking at a fat, oil, or wax. Proteins are a little different. They have a wide variety of polymer type structure than when they make those big molecules, but it doesn't matter what protein we're making. The building block of a protein is an amino acid. So amino acids make up the base of a protein. The last one here are the nucleic acids. When we put them big together, we still call them nucleic acids, and these are going to be things like DNA and RNA. Okay, and DNA and RNA are really important to living things in cells. But the building block for DNA and RNA are the nucleotides. Okay, so nucleotides are the building blocks of nucleic acids. Now, there are two types of chemical reactions we want to talk about when it comes to these polymers. We can either build them or break them. Okay, so synthesizing them is when we build these polymers, and degrading is when we break them down. So the first example here is when monomers are joined together to form the bigger molecule, the polymer. This is going to be known as dehydration or a synthesis reaction, and we are going to do this by removing water. So synthesis is the construction type of chemical reaction. And when we build something, remember, we're going to have to put energy into it. So there's going to be an input of energy here. Water is going to end up being generated here because we're going to remove it from the molecules so that they bond together. And this is why it's called dehydration, because we're removing it from the molecules. So let me kind of show you a kind of picture of this. The reactants that are going in are two monomers. So I have a monomer plus a monomer. And you'll notice with this dotted line, I have circled the part that's going to make water because water is H2O. So see how there's two H's and an O here. What we're going to do is we're going to take that out, and this is then going to create a polymer because those monomers have merged together. You'll also know that that water comes out on the other side because we removed it. Since we're synthesizing this reaction, energy has to go into it. Okay, so there's an input of energy to build something. So over here, if we look at dehydration reactions, we're going to start with the building blocks, the monosaccharides, and we are going to bond them together by removing water. This creates polysaccharides. If I have fatty acids plus glycerols putting together, I end up with lipids. A chain of amino acids forming together forms a protein, and then the nucleotides coming together make the nucleic acids. Now, on the other hand, if I want to take a big molecule, a polymer, and I want to break it into pieces and make it into those monomers, those building blocks, I'm going to add water to do this. This is known as hydrolysis. Hydro means water, lysis means to break. So I'm going to add water to break the bonds, kind of like when you play Red Rover and you break that bond between the two people holding hands. So these are known as decomposition or degradation reactions, and energy is going to come out of them. So energy is going to be released. So if we use the kind of similar example we had on the last page over here, we have a polymer. We are going to break it apart by adding water. This causes energy to be released, and we break the water apart. So we have H on one molecule and OH on another. Now, a big concept here, guys, is for most chemical reactions, this process of either building something or breaking them down, a lot of times requires an enzyme. We're going to talk about enzymes in a future video, but they actually make the reaction go faster. And these types of enzymes are known as being a catalyst. So going back over here, we started with the polymer, and now we're going to break it down into the monomers. So if I start with polysaccharides and I add water, I'm going to end up with monosaccharides. If I have lipids and I add that water and break it apart, I'll get fatty acids and glycerol. Proteins will be broken down into amino acids, and nucleic acids will be broken down into nucleotides. Now remember, these are covalent bonds that we are breaking at this particular point, okay? So we're adding water to break those covalent bonds. Now, foods contain carbohydrates, lipids, proteins, and nucleic acids. And these subunits are used for the building blocks that our cells use for energy. And so we don't necessarily use the proteins directly that come from our food. We break them down into those amino acids, and then we build the proteins that we need. Okay, so there's a constant back and forth here with this idea of building molecules and breaking them down. And so when we look at nutritional labels, you'll notice that these things are present. It'll show you the total fat, total carbohydrates, and proteins. And if there are more than 5 grams in them, there is a lab that we can do or experiment where we can add things to them and they can actually detect that those molecules are present. Okay. And so reading nutritional labels can be important in helping us understand if we're getting what we need from the carbs, lipids, and proteins that we are consuming. So we're going to look at each of these macromolecules in a little more detail. And we're going to start with the carbohydrates. Well, guys, carbohydrates kind of get a bad rap when it comes to diets and things like that because people always want to cut them out. And the reason being is that your cells, they're kind of lazy and they prefer the quickest and easiest way to get energy. And carbohydrates are the way to do that. So carbohydrates have a general formula of a carbon matched with 2 hydrogen and an oxygen. And a lot of times we have it in parentheses with an N because that N can be any kind of number. So in this particular case, that N could be 6. And if that's the case, then it would be C6 because C was a 1. So 6 times 1 is 6. H12 because 6 times 2 is 12, and O6. This is known as the monosaccharide for hexose sugars. Hexose means that they have 6 sides to them. Simple sugars are the sweet stuff. They're the stuff that's really sweet and we can consume them and break them down very quickly. And this is because they only have 1 sugar molecule. These hexose sugars, one of the best examples of this is glucose. And glucose is your sugar of choice for your cells. And the reason it is it's an immediate energy source for your cells. Okay, and so it can give it fuel very quickly. Now, when we look at this, your cells being able to utilize the sugar does rely on oxygen, and we call this cellular respiration. Now, they get their name as hexose sugars because they have a hexagon type shape. This next one are called the pentose sugars. These normally are C5H10O5. They have 5 sides to them. These sugars are really important in the building block of nucleotides. Nucleotides are going to have ribose sugar if they are RNA nucleotides, and they're going to have deoxyribose sugar if they are DNA nucleotides. If you'll notice, glucose, ribose, deoxyribose, they all end in ose, and so a good rule of thumb is if it ends in ose, it is a type of sugar. Now, disaccharides are also a very simple sugar, but they have 2 monosaccharides bonded together, so they're a little more complex than the monosaccharide. There's some examples here. One of the main examples is sucrose. Sucrose is a table sugar and it comes from plants. Okay, so this type of sucrose, when we look here, is a glucose that is bonded to a fructose. So water has been removed in order for them to bond together. Lactose is the sugar that is found in milk, and this is going to be where we have a galactose that's bonded with a glucose. And so it gives it a different kind of set of properties. One thing about this too is if somebody is lactose intolerant, they do not have the enzyme that is used to break this sugar down. Okay, so they are missing the enzyme that in their body that would break this down as they digest it. And this is why they end up having a lot of discomfort, pain, things like that when they eat anything that has this sugar present. Polysaccharides are going to be the polymers of a bunch of monosaccharides put together, and these are very complex sugar. And guys, this could be thousands of glucoses bonded together and things like that. In plants, there are 2 types of polysaccharides that are present. One is cellulose, and cellulose is a structural type of sugar. It's what the cell walls of plants are made out of. So when we talk about their plant cells, their cell wall is made out of cellulose. Starch, on the other hand, is the sugar that they store. So they put a bunch of sugars together, store it up for use for later. And they do this by completing photosynthesis to make these sugars. Guys, this is like in a potato. When you pull that potato out, you are taking the energy source of that particular plant. In animals, we also see 2 types of polysaccharides. So the first one here is chitin or chite. This is a structural type of carbohydrate like we saw with cellulose, but this makes up the exoskeleton of things like crustaceans, so like crabs, lobsters, shrimp, that sort of thing, as well as bugs like insects. Okay, so that really hard part that crunches, that's made out of chitin or chitin. We also have glycogen. Glycogen is the temporary storage for energy, and we find this is stored most of the time in our livers. So we do store energy away for later, and we do store it in our liver as glycogen. Now again, a lot of these may end in ose like cellulose, but some do not when we're talking about polysaccharides. So a quick little overview here of the carbohydrates. Over here we start with the monosaccharides. Monosaccharides can be bound together through synthesis to make disaccharides. More disaccharides can be bound together to make polysaccharides. However, we can also break these down for decomposition where we go from polysaccharides to disaccharides to monosaccharides. So these are reversible reactions. They're constantly going back and forth in our bodies. So other examples of carbohydrates are going to be with monosaccharides, you have ribose and deoxyribose. For disaccharides, you have sucrose and lactose. And for polysaccharides, you're going to see that we have cellulose, chitin, and glycogen. Now guys, under here I talked about how monosaccharides and disaccharides are considered your simple sugars, and they do all end in ose. Whereas your polysaccharides are your complex carbs or complex sugars. So which complex carbohydrates are structural in function? If we're talking about the structural component of these particular sugars, we're going to see things like cellulose in plants and chitin or chitin in animals. All right, so this brings us to lipids. Lipids are our fats, and one of the main components or characteristics of lipids is that they are insoluble in water. They are hydrophobic. They do not like water. Now, they are soluble, however, in alcohol. Now, fats and oils are the basic monomer of our lipids. Okay, so when we look at this, there are fatty acids and glycerols binding together to make your fats, oils, and even your waxes. Now, with this, they can be also known when we're talking about fatty acids and glycerols, they can be known as triglycerides. So tri means three. So this means there are three fatty acids, and the glyceride tells you there's a glycerol present. So I've got three fatty acids attached to a glycerol. When we look at fats, one of its main jobs is for long-term energy storage. Guys, fats and lipids contain 3 times more energy than glucose does. However, they're harder to access, so your cells would prefer the sugar. Another thing that fat does for us is it provides insulation. Thus, in these pictures you see down here with a polar bear and a walrus and the penguins, they have a lot of what we could consider blubber or fat that's present, and it's for insulation. We also have quite a bit of this that surrounds certain organs, and we do need some of it there because it acts as insulation and cushion for those organs. Now, with fats, you've probably heard of something called saturated or unsaturated fats, but you probably maybe didn't know the difference. So let's talk about the difference here. This first set of fats are your saturated fats. These are associated most of the time with animal fats. They are more solid at room temperature, things like blubber, lard, that sort of thing. So these are going to be harder for your body to process. So we should try to reduce them in our dietary intake. It does not mean cut them out completely, but we shouldn't be using them all the time when we cook. And the reason being is they are associated with heart disease. Now, the reason they are considered saturated, it means that all of the carbons are completely saturated with bonding with multiple molecules. There are no double bonds with the carbons. Okay, so double bonds mean we have the 2 lines between them. And if you look here, these are all carbons with a single line between them. So they form a single set of bonds with each other, and then they're going to bond with other things. And a lot of times they are also going to be completely covered with hydrogen, and that helps you be able to visualize that they are saturated as well. On the other hand, if they're unsaturated fats, they are associated more with plant oils. They're more liquid at room temperature. This is like corn oil, peanut oil, olive oil, avocado oil, all those types of oils that you can buy at the store. These are better fats for your diet. Your body can actually process them a lot easier due to that idea of them being liquid. And also, they do form those double bonds. So if you look here, there's going to be where there are a double bond between the carbon, which means those carbons cannot bond with multiple things more than what they had before because they have a double bond between them. So this right here is showing you a triglyceride. We said triglycerides have 3 fatty acid chains, which you can see here in gray, and they're bonded to a glycerol molecule. Now, the big thing here is we need to determine if they're saturated or unsaturated fatty acid chains. So if you look at the first one, you can see that there are no double bonds present. Since there's no double bonds present and they are saturated or completely covered in hydrogens, we would say that that one is a saturated fat. In number 2, we also see the same thing. There are no double bonds between carbons. And so because of that, it is also a saturated fat. But in this last example, you can see there is a double bond. And if you'll notice, that double bond also causes the chain to bend a little bit. This is because there's 2 hydrogens also missing at the bottom. And so this one is considered an unsaturated fat. So another thing to note on this is that a triglyceride can actually be a combination of saturated and unsaturated fatty acids. The next type of fat I want to talk to you about are waxes. Waxes are going to have long fatty acid chains, but they're also going to have some of those alcohol chains or functional groups that are present. Now, the function of waxes are to help waterproof areas. Plants and animals will use this to waterproof certain structures. This is going to help with it being protective in that sense. And so earwax is one of those for us. Now, don't be like this guy and clean out his ears with Q-tips for earwax. That actually can compact it more and cause more problems, right? But we do see that it is a protective function. We also see that bees will do this with wax and help protect their honeycombs. And we also see there's a waxy kind of film on plant leaves that they call a cuticle. This is to help protect the leaf from water being lost, okay, from inside the leaf out. And so this is why water droplets will kind of pool and not really stick too much to the leaf. The next fat we want to talk about are phospholipids. The structure of a phospholipid is a fatty acid and a glycerol, but it's bound to a phosphate group. All right, and so if you look here, we see that there are 2 fatty acid tails that are going to be attached to this phosphate group. Now, this creates 2 regions on a phospholipid. Now, this creates a polar head of that phosphate functional group, and this actually makes it hydrophilic. It likes water. However, the tails, because they are made out of fatty acids, they do not like water, so they are hydrophobic. This creates a very important structure because we have one side liking water and one side not. And so it makes this double layer, a bilayer that you can see here. And this is actually what makes up that plasma membrane of our cells. And it acts as a barrier where water can be outside the cell and inside the cell, but we have a barrier in between. So again, water outside and water inside, creating a barrier. Okay, and so the head part likes the water, the tails do not. The last type of fat I want to talk to you about is steroids. Steroids are fused carbon rings that actually look kind of like honeycomb. These fused rings provide more structure, and they provide structure to things like the plasma membrane. So cholesterol is the main one that's found in your plasma membrane in our animal cells, helps provide structure within that membrane, and they are important. Now, you may have heard cholesterol before if you have high cholesterol, but you cannot cut cholesterol out completely because your body does need it. On the other hand, we also see cholesterol is the kind of precursor to our sex hormones like estrogen and testosterone. And steroids tend to get a kind of a little bit of a bad rap when we talk about anabolic steroids, when you're taking them when you shouldn't to try to make yourself stronger and perform better, like in sports and things like that. Next, we want to talk about proteins. Proteins, the building blocks are going to be amino acids. Amino acids are going to form together when we put one monomer combined to another monomer and so on. And these are going to be called polypeptides because the bond that happens between each amino acid is known as a peptide. So here is the amino acid structure. You can see that it does have the carbon in the middle, but it has an amino group on one side and a carboxyl group on the other. We also see that there's an R group there that's a variant group. These variant groups are going to help create 20 different amino acid types. The different combination of these amino acids put in a sequence help us create lots of different types of proteins. So amino acids will form together to make peptides. Peptides will form together to make polypeptides, and then eventually they'll fold to make a 3D protein. So there are 20 different amino acids. That R group can be simple like an H, like you see in glycine. These are all highlighted, which are the parts that make each of them different. Or they can be a very complex chain like you see with arginine down at the bottom in blue. And so it just depends. Each of those give those different amino acids different properties. Okay, so they are named based on that shaded area. Now, you do not need to memorize these 20 different amino acids. The big thing is knowing how to use a chart for them when we start talking about how proteins are made later on. Now, the special kind of bond that happens here, because we're removing water, that is formed between amino acids is known as a peptide bond. So the special bond that happens here is called a peptide bond. Okay, so water is formed as a product, and we see that we are going to form this new element or this new molecule. All right, so now going back to some of the information we talked about earlier in this particular video, what kind of chemical reaction is this? If I'm taking two parts and I'm going to fuse them, bind them together, this is obviously a synthesis reaction. Because we're also going to be building something and water was removed, it is also known as dehydration. Now, proteins can have lots of different types of functions, and this obviously is going to be related to the different ways that they're put together structurally. But some of these proteins are going to help with support. They're structural proteins like keratin in your hair or your fingernails. They help give you that structure. Collagen in your skin, which helps keep it really tight and nice, as well as ligaments and tendons. And even the silk that's found in the webs of spiders is made out of protein. Other proteins are for metabolic purposes. These are going to be things like enzymes. We talked briefly about these, but enzymes speed up chemical reactions and help them go faster, whether it's to build something or break it down. Also, proteins can help in transport. Transport, like hemoglobin in our blood, transports oxygen. As some plasma proteins will act as like a bridge for transport between the inside and outside of your cells. Some proteins act as defense. You've probably heard about these types of proteins before. They're called antibodies. Some are regulatory, meaning they're things like hormones, as well as some help you with motion, and these are going to be found in your muscles called actin and myosin. Now, in order for each of these proteins to do whatever job that they are given, their shape is very important. They end up having what we call this 3D type of shape that allows them to perform their job. Now, in order to get to that shape, there are going to be a series of structural steps. So step one is the primary structure. The primary structure, guys, is a chain of amino acids. Each of these circles is an amino acid, with the little line being a peptide bond. So if you notice, they look actually kind of like a string of pearls. So the primary structure looks like a string of pearls. When we take these pearls and we actually kind of wind them up in a helix, or we fold them where they look kind of like a beta sheet or pleat, this is known as the secondary structure. So the secondary structure is going to be where we start to wind them or fold them in different ways. Now, this winding and folding is going to depend on those functional groups found on each of these amino acids. Then this leads to the tertiary group. The tertiary group is where we actually get more of that 3D shape. It almost looks kind of like a glob, so they call it the globular shape. It looks kind of like it's just been wadded up and messed up, but there's a purpose and actual order to how it folds like this. Now, some proteins are actually functional at the tertiary phase. Others have to go to the fourth phase, which is the quaternary structure. Now, the quaternary structure is where we take multiple tertiary or globular proteins and we fuse them together. In this case, you have two of the pink and they are merged with two of the purple to form this particular structure. Okay, now again, proteins are only functional in their tertiary or quaternary stage. If they unfold, we've got an issue. This unfolding of a protein is called denaturation. It's where it loses its normal shape, so it's no longer functional. Because, guys, structure is related to function. So change in shape, change in function. Now, these bonds have been altered or broken somehow, and they can end up be broken or altered by things like pH, heat, heavy metals, salt concentration. All those things could ultimately affect a protein. So the protein goes from being normal to being unwound or denatured. Okay, so tertiary shape, because remember I told you it could be functional in the tertiary, and it unfolds back to either its primary or secondary shape. Now, if denaturation is too severe, the protein may never regain its structure or its function, and we'd have to start over with making new ones. An example here is like with eggs. Okay, in eggs, there is a big set of proteins called albumin. This is one reason why they're a good source of protein. The thing is when you cook an egg, due to the high temperature, it changes the albumin, which is the white part of the egg, from being this kind of clearish yellow, light yellow color to a white. You have denatured and unfolded all those proteins to where they can no longer function. And this is permanent. You can't go back to the way it was before. Okay, so sometimes it's a permanent change. All right, the last group I want to talk to you about are the nucleic acids. These are deoxyribonucleic acid, DNA, and ribonucleic acid, which is RNA. Now, DNA, guys, is your hereditary material, okay? And it is the hereditary material for all living things on planet Earth. Whether it is a plant, whether it's a sponge, a bug, a fungus, like a mushroom, we all have DNA. The structure and its relationship to ribonucleic acid, RNA, which is DNA's helper, RNA is going to help us make the proteins that we need, depending on what organism we are. Now, the building block of a nucleic acid is a nucleotide. So this nucleotide is going to have three parts to it. It's going to have a phosphate group shown here in yellow. It's going to have the pentose sugar, the 5-carbon sugar that's shown kind of in this lightish green, and then a nitrogen containing base. Okay, now this nitrogen containing base in this case is marked with a C. However, it could also be marked with an A, G, T, or U, depending on which type of nitrogen containing base it is. In this case, with it being C, this is known as cytosine. Now, the nucleic acid is the polymer of the nucleotide. So we're going to have a bunch of these nucleotides bound together in order to make a nucleic acid like DNA or RNA. So let's take a little closer look at DNA and RNA, as well as the last one I want to talk to you about, which is ATP. All right, so DNA is known as deoxyribonucleic acid, and it does have a pentose sugar called deoxyribose. That's its name. The nitrogenous bases that can be present in DNA are A, G, C, and T. So adenine, guanine, cytosine, and thymine. There's also going to be one phosphate per nucleotide. So there's only going to be one per nucleotide. The standard type of structure of DNA is a double helix, meaning it looks like a twisted ladder. Now, obviously in this picture, it's not going to look that way because it's like we unwound the ladder so that you can see it. We have two strands that are bonded together in the middle with hydrogen bonds. These hydrogen bonds are between molecules like adenine and thymine, so A and T, or guanine and cytosine, G and C. Now, the purpose of DNA is it's going to store your genetic information, and this genetic information is going to act as the code to help you build proteins. On the other hand, when we look at ribonucleic acid or RNA, we do have a ribose sugar instead of deoxyribose. We are going to see there is going to be one difference in those nitrogenous bases. Instead of T, you're going to see U. So they have adenine, guanine, cytosine, and uracil instead of thymine. The phosphate group is also one per nucleotide. Now, RNA only comes in single strands. Okay, so there's not a double strand here, but you'll notice that the structure is very similar with that nucleotide. We see that it forms a sugar phosphate backbone. So it goes sugar, phosphate, and we see that both in DNA and RNA. Now for RNA, this is going to carry out the actual process of protein synthesis, the making of proteins. RNA is critical in that process. Now, ATP is known as adenosine triphosphate. So this means it's going to have an adenine group. Okay, that's where the A comes in. And then the tri tells you there are 3 phosphates that are present. The sugar that's still here is ribose sugar. We have only A for adenine, and we're going to see that there are 3 phosphates. So this is what ATP looks like. We have adenosine, the A, with ribose sugar and 3 phosphates. These 3 phosphates bound together are going to help us have energy. So ATP here is going to be the energy carrier of our cells. This is the currency, like the money in that sense, for energy in your cells. Now, the reason why these are so important is you have your basic flow of genetic information. We have DNA containing the genetic information or the instructions that's going to then be formed into RNA. RNA is going to then go through the process of making a protein. This right here is known as the central dogma, and we will talk more about it, just like we'll talk more about ATP in future videos. But guys, the gene on the DNA that has the code is going to be a particular segment on that base that's going to make the RNA. The genome, all of the genes in your DNA, is going to be what makes you, you. Now, if any of these gene areas get mutated or changed, we see that it could cause a problem in what kind of proteins being formed. And an example here is sickle cell disease. And like I said, we will talk about this more in future videos. So if you have any questions or concerns, please don't be afraid to ask.